Good afternoon. I am Dr. Sucheta, Associate Professor in School of Medical and Allied Sciences, here Mangalam University. Today I am going to present my lecture on parasympathomimetic agents. The content of this lecture is cholinergic neurotransmitters, biosynthesis, catabolism of acetylcholine, then the different type of cholinergic receptors and their distribution. Then the parasympathomimetic agents, their structure-activity relationship. Finally, we will discuss the classification of parasympathomimetic agents, direct acting and indirect acting. Now, the biosynthesis of acetylcholine. Acetylcholine is a neurotransmitter, which is a sympathetic neurotransmitter, and it is biosynthesized, that is inside the body, with the help of enzyme choline acetyltransferase from choline and acetyl coenzyme A. And this happens in the cytoplasma of the nerve endings, nerve terminals. The choline, which is used in the synthesis of this acetylcholine, is produced from the hydrolysis of acetylcholine by the enzyme acetylcholine esterase. And this acetylcholine, which is released in the synaptic region, which further get hydrolyzed by this enzyme acetylcholine esterase into the choline and acetic acid. That choline is recaptured or reuptake by the presynaptic terminals by the high affinity uptake system under the influence of sodium ions. And this uptake system is inhibited by quaternary ammonium bases like the hemicholinum, 2-hydroxyethyl triethyl ammonium. Whereas this choline not only produced by the hydrolysis of acetylcholine, but it can be synthesized in the liver through a amino acid serine, and that serine is converted into ethanolamine, which further undergo the methylation process with the help of enzyme methyltransferase. Now, after the formation of acetylcholine, it is not released in the directly in the nerves, but they are stored in the nerve terminals in small vesicles in the form of a complex, and that complex is acetylcholine plus ATP plus prostaglandins and a soluble acetic proteins, that is vesicolin. And this complex is very much stable. Why they are stored in this complex form? Because it prevents it from the hydrolysis by this esterase enzyme, acetylcholine esterase enzymes. And 1000 to 50,000 acetylcholine molecules are stored per vesicles, and one nerve terminal has 3 lakhs or more vesicles. After this, these vesicles, whenever some stimulus is given, then these vesicles they get binded with the nerve terminals, and by the process of exocytosis, they releases acetylcholine in the synaptic region. After that, there are two points: whether they bind with the receptors, or finally they get acetylcholine molecules get hydrolyzed by this esterase enzyme into the acetylcholine and acetic acid, and by this way the action is terminated. Now, how many types of cholinergic receptors are there in our body, and where they are located, and what the biological action is shown when the acetylcholine bind with these type of receptors? There are basically two types of receptors: muscarinic receptor and nicotinic receptors. Muscarinic receptor is further of 5 types: M1, M2, M3, M4 and M5. Whereas nicotinic receptors of 2 types, NM and NN. First, the muscarinic M1 receptor is present in various secretory glands like gastric gland, lacrynal gland, salivary glands, and in the stomach also. The gastric juices are secreted only after stimulation of these glands. It is also present in this cerebral cortex region. And when the acetylcholine bind with the M1 receptor present on the secretory glands or the cerebral cortex, they causes the stimulation of these glands, and they causes the release of the secretions, like they enhances the salivation, stomach acid, sweating, and they causes the lacrimations. The second type of receptor, muscarinic receptor, that is M2, which is present on the heart, smooth muscle, cardiac muscles. Once the acetylcholine bind with the M2 receptor present on the heart, they decreases the heart rate. That is, this decrease in heart rate is known as the bradycardia. M3 receptor is present on all the smooth muscles which are not in our control. They are contracted by their own, So when the acetylcholine molecule bind with these smooth muscles, which is present on the GI, gastrointestinal tract, urinary bladder, respiratory tracts, they causes their contraction. And because of their strong contraction, they causes diarrhea, bronchospasm, urinary bladder contractions, and they also causes the contraction of pupil. And the process of contraction of pupil or ciliary muscle is known as the meiosis. That's why by this contraction they causes the flow of aqueous humor, and that's why they are used, these analogs or cholinergic drugs. They are used in the treatment of glaucoma, open angle glaucoma. Now the M4 and M5 type of receptors, which is present on the hippocampus and substantia nigra, they are involved in the mood, cognition, and the drug seeking behavior. NM receptors, NM receptor is present, M indicate the muscle. It means a nicotinic receptor which is present on the skeleton muscles and plates. So they causes the contraction of skeletal muscles, and N receptors are present on the neuron, that is neuronal receptor subtypes, and they causes the secretions of epinephrine, which controls the ANS. Now, as the chemistry point of view, we should know about the SAR. SAR means structure, activity, relationship. It means once the structure is changed, the activity automatically gets changed. So where the change in structure is beneficial and where if we do the changes in the structure, that is not fruitful. So if we divide this is the acetylcholine molecule, and if we divide this whole molecule into the 4 different parts, one is the quaternary ammonium part, which contains the positive charge, and it is known as the quaternary ammonium head or cationic head. The second is the ethylene linkage, which is helpful in binding this quaternary ammonium nitrogen with the ester. The third is the acetyl part, COOCH3. This COOCH3 is the acetyl part, and this acetyl part contains the fourth part, that is the ester group. So by doing the modification in different parts, that is these 4 parts, alteration in the quaternary ammonium head, then doing the changes in the ethylene linkage, doing the changes in the acetyl group, or doing the change of an ester group, we get the different compounds. Some compounds are beneficial and some compounds are not so much fruitful, and some show very less activity, which are not of any use. Now if we take one by one, first one is alteration and modification of quaternary ammonium head, because it contains the positive ions, so that which makes it a cationic head, that is required for the optimum activity. It means it is responsible for binding with the anionic part of the receptor. So if the ammonium part, that ammonium part, quaternary ammonium part, is replaced by onium group like sulfonium group, phosphonium group, arsonium groups, 90% of activity get lost. So we can't replace this ammonium from other by other onium groups. Only the 10% activity will be retained. If one of the methyl group of the trimethyl ammonium group is exchanged by the higher alkyl groups like ethyl, propyl, butyl, and all others, then the 75% activity is lost. It means only the 25% activity will be retained. So this is also not a fruitful result. Next one is if we replace more than 2 methyl group by the higher alkyl groups like ethyl, propyl, almost complete loss of activity was observed. And if we replace this nitrogen with the carbon, it becomes uncharged because carbon has a 4 valency and the compound is 3,3-dimethyl butyl acetate. And this produces the activity that is 0.003%, which is negligible. Now the distance between the ammonium group and the ester group. This ethylene chain assures the proper distance. Distance between the onium and ester group, that must be 0.6 nanometer. And it follows the rule of 5. If we increase this chain length, it is 2, that is ethylene linkage. If we increase the chain length up to 3, 4, 5, that activity will get decreased. So the rule of 5 says that there should not be more than 4 atoms between the quaternary nitrogen and the terminal methyl groups. Further increase in the chain result in decrease in activity. But the one thing which we find very good, that is branching. If we do the substitution in alpha carbon or beta carbon, we get a fruitful results. At alpha position, the methyl substituent that produces the methacholine, which is more potent than the acetylcholine, and it shows the more potent muscarinic effects. But it is a very weaker nicotinic action showing compound. Whereas the alpha position, if the methyl group is at alpha position, it shows the more nicotinic effect, not muscarinic effects. And the S isomers of methacholines is much more active, and R isomer is 1/240th as active as that of a acetylcholine. Now, if we replace this acetyl group by other acyl groups, the most important or the most we can say that good results we obtain from this is carbamate. If we replace this acetyl by the carbamate, COOCH3 is replaced by COONH2. This NH2 makes it more potent, and these carbamates are known as carbachol. So carbamol derivatives like carbamate esters of choline, carbachol, which are highly active, and because of why they are active, because this carbamate NH2 COONH2 makes them resistant to the hydrolysis by esterase enzyme, acetylcholine esterase. That's why they remain longer for longer period of a time in the body, and they show good effects. Now the beta choline, which is a carbamate ester of beta methylcholine. The difference between carbachol and methacol is that is methyl group. This methyl group at the beta position. This beta-chol is much more active than the carbachol. So that means that methyl substituent at beta position makes it more potent. Now replacement of ester group by other groups. Yes, ester group, which is the last substituent, fourth part of the acetylcholine. If we exchange this or we replace this ester group by the keto group, ether group, or other groups, we get good results. The keto compounds, they are very potent, but they show little muscarinic action. But yes, they are active compounds, and they are stable and potent compounds. And if we can say that reverse the position of ethyl and carbonyl oxygen, we get good compounds which show the strong muscarinic and moderate nicotinic activity. This was all about the SAR, but SAR get never completed until unless we do the stereochemistry of the compounds. Stereochemistry of acetylcholine, the S isomer is active, R isomer is inactive. So the S or plus trans enantiomer is much more active, and we know that this acetylcholine, they exist in the synclinal, anticlinal, and the antiplanar. Now the classification of cholinergic drugs. The classification, basically the cholinergic drugs are of two types: direct acting, indirect actings. Direct actings mean choline esterase, whereas and natural alkaloids, whereas the indirect actings means which are not binded with the receptor directly, but they increases the concentration of acetylcholine in the body by inhibiting its hydrolysis. So the direct acting cholinergic drugs are the drugs which show the same structure as that of an acetylcholine, and instead of binding with the acetylcholine, they bind with the receptors and show the same effect as shown by the acetylcholine. That's why they are also known as the mimetic drugs, cholinomimetic drugs, or the cholinergic drugs, or the parasympathomimetic drugs. All are the synonyms. So choline esters में carbachol, methanicol, methacholine, bithanicol, all they come under the choline esters. Whereas the pilocarpine is a natural alkaloids obtained from the leaves of a plant. The indirect acting compounds, basically they are of two types: reversible cholinesterase inhibitors or irreversible cholinesterase. Reversible are those like physostigmine, neostigmine, pyrostigmines, whereas irreversibles are isofluranoids, parathione, malathiones, and all that. Now, if we discuss all these in detail, directly acting cholinergic drugs directly bind with the receptor, indirectly they do not bind with the receptors, but they increases the concentration of acetylcholine inside our body because they inhibit its hydrolysis. If it is not hydrolyzed, automatically its concentration will get increased, and it itself will get bind with the receptor and show its effects. So these pilocarpine obtained from the leaves of shrub Pilocarpus jaborendi, and it crosses the blood-brain barrier. It has a muscarinic action and very less nicotinic actions. They are used in the open treatment of the open-angle glaucoma, in the treatment of midriatic, which is produced by the atropines, and it breaks the ions between the iris and the lenses. They also increases the salivary secretions. These are the structures of direct-acting cholinergic drugs. Acetylcholine, which we have already discussed in the SAR. This is quaternary ammonium, ethylene linkage, and the ester part. Carbachol and ethacholine, the difference is only the carbamate group. This CH3 group is replaced by the NH2 groups. Then bithanicol and carbachols are same, but only difference is the methyl substituent at beta position. Methacholine and acetylcholines are the same, only the methyl substituent at beta position. Now, pilocarpine is a naturally occurring alkaloids which is obtained from the Pilocarpus plant leaves. Indirect acting reversible cholinesterase inhibitors, it means that they inhibit the enzyme by acetylating its hydroxy part of the serine, residue of the acetylcholinesterase. So acetylation process is a reversible. The enzyme get regenerated, and there are drugs like neostigmine, pyridostigmines. These are having quaternary ammonium group. Hydrophonium chloride. This is also having the quaternary ammonium. Physostigmine does not contain any quaternary ammonium. Ambinonium is a biquaternary salts. Tacrine. Tacrine is a novel drug which is now a time of latest drugs used in the treatment of Parkinson's. Irreversible cholinesterase inhibitors, organophosphates, which are used as insecticides, they are highly toxic, like palathion, malathion, and the isoflurates. These are the structures. Neostigmate, again, it contains the quaternary ammonium. Pyridostigmate also contains the quaternary ammonium. Yes, physostigmate does not contain the quaternary ammonium. This ambifonium is a biquaternary salt. Hydrophonium is also containing the quaternary ammonium. Tacrine is somewhat having different structure as that of all other drugs which come under this category. Irreversible, these are the isoflurate, all the phosphate, organophosphates, ecothiopate, iodide, malathion, and parathiones. They phosphorylate the enzyme cholinesterase and irreversibly inhibit it. It means that they are not reversible one, and we can't get the cholinesterase enzymes back after the decrease in concentration of these drugs. So this is irreversible. So that's why they are highly toxic, and in the Second World Wars, nerve gases they are used. Thank you very much.